Hello, my name is Joshua Mugabe. I welcome you to Non Mendelian Inheritance. According to Mendel's law, phenotypical characteristics would be determined by a pair of factors, alleles, that separate independently in gametes. What are the main types of inheritances that are exception to Mendel's rules? There are many types of inheritance that do not follow Mendelian pattern. Notable among them are multiple alleles, gene interactions, complementary gene, epistasis, and quantitative or polygenic inheritance, linkage with or without crossing over, sex-linked inheritance. Pleiotrophy, lacking of dominance, lethal genes do not fit as variation of inheritance since genes have the same behaviors and at the same time obey Mendelian's law. Mutation aneuploidies are abnormalities that also alter Mendelian's pattern of inheritance, as well as mitochondrial inheritance, passage of mitochondrial DNA from the mother through the cytoplasm of the egg cell to the offspring. What is the genetic condition in which heterozygous individuals has different phenotype from the homozygous individual? The condition is called lack of dominance, or it can happen in two ways: incomplete dominance or codominance. In incomplete dominance, the heterozygous present an intermediate type between the two types of homozygous, as in sickle cell anemia, in which the heterozygous produce a sickle red blood cell and some normal red blood cell. Codominance occurs, for example, in genetic determination of MN mobile blood group system, in which the heterozygous has a phenotype totally different from the homozygous of being an intermediate form. What is pleiotrophy? Pleiotrophy or pleiotrophy is a phenomenon in which a single gene conditions several different phenotypic traits. Some phenotypical traits may be sensitive to a pleiotrophic effect, For example, inhibition of other genes, even when conditioned by a pair of alleles in a simple dominancy. In this case, a mixture of pleiotrophy and gene interactions is characterized. What are lithogenes? Lithogenes are genes having at least one allele that, when present, the genotype of an individual causes death. These are recessive lithoalleles and dominant lithoalleles. There are also genes having alleles that are dominant when in heterozygosity, but litho when in homozygosity. For example, the dominance related to the phenotype does not correspond to the dominance related to lethality. What are multiple alleles? Is there dominance in multiple alleles? Multiple allele is a phenomenon in which the same gene has more than two different alleles. In Non Mendelian inheritance, a gene has only two alleles. Obviously, these alleles combine in pairs to form the genotypes. In multiple alleles, relative dominance among the alleles may exist. A typical example of multiple allele is the inheritance of the ABO blood group system, in which there are three alleles, A, B, and O. Hemoglobin A, hemoglobin B, and hemoglobin O. Hemoglobin A is dominant over hemoglobin O, which is recessive in relation to other hemoglobin B allele. Hemoglobin A and hemoglobin B lack dominance between themselves. Other example is the color of rabbit fur, conditioned by four different alleles: C, CCH, CH, and C. In this case, dominance relations are C is greater than CCH, which is greater than CH, which is greater than C. Symbol that greater than means dominates over. What are gene interactions? What are the three main types of gene interactions? Gene interaction is a phenomenon in which a given phenotypical trait is conditioned by two or more genes. Do not confuse with multiple alleles, in which there is a single gene having 3 or more alleles. The 3 main types of gene interaction are complementary genes, epistasis, and polygenic inheritance, or what we call quantitative inheritance. What are complementary genes? Does this inheritance pattern obey Mendelian's second law? Complementary genes are different genes that act together to determine a given phenotypic trait. For example, consider a phenotypical trait conditioned by Z complementary genes, whose alleles are represented X, small x, Y, and small y. In F2, 4 different phenotypic forms are obtained. X dash Y dash double dominant. X dash small y dominant for the first pair, recessive for the second. Then X small x and capital Y dash, recessive for the first pair and dominant for the second. And small xx and small yy double recessive. This is what happens. For example, regarding the color of Badgaliga feathers, in which dominant double interaction result in green feather. Dominant fur, the first pair, recessive for the second interaction results in yellow feathers. Recessive for the first pair, dominant for the second pair interaction leads to blue feathers. And double recessive interaction leads to white feathers. Each complementary gene segregates independently from others, since they are located on different chromosomes. Therefore, the pattern follows Mendelian's second law, although it does not obey Mendel's first law. What is epistasis? What is the difference between dominant epistasis and recessive epistasis? Epistasis is the gene interaction in which a gene, the epistatic gene, can prohibit the phenotypic manifestation of another gene, the hypostatic gene. In dominant epistasis, the inhibitor allele is the dominant allele. For example, capital I of the epistatic gene is so inhibition occurs in the dominant homozygosity, capital I, or in heterozygous or heterozygosity, capital I, small I. In the recessive epistasis, the inhibitor allele is a recessive allele of the epistatic gene, small I. So inhibition occurs only in recessive homozygosity, small I, small I. In the hybridization of the two genes, 4 different alleles, 2 of each pair, how does epistasis affect the proportions of the phenotypic forms in the F2 generation? In dihybridization without epistasis, double heterozygous parent individual cross in F2, 4 phenotypic forms appear. The proportion is 9 double dominant, 3 dominant for the first pair, recessive for the second pair, 3 recessive for the first pair, dominant for the second pair, and 1 double recessive. 9 to 3 to 3 to 1. Considering that epistatic gene is the second pair and that the recessive genotype of the hypostatic gene means lacking the characteristic, in F2 generation of the dominant epistasis, the following phenotypic forms would emerge. (13) dominant for the second pair or recessive for the first pair. The characteristic does not manifest. 3 dominant for the first pair, recessive for the second, i.e. the characteristic manifests. Phenotypical proportion would be 13 to 3. In recessive epistasis, in F2 phenotypic forms that would emerge are 9 double dominant, characteristic manifests, 7 recessive for the first pair or recessive for the second, i.e. the characteristic does not manifest. So the phenotypic proportion would be 9 to 7. This example shows how epistasis changes the phenotypical forms and proportions from the normal 9 to 3 to 3 to 1 in F2 to 13 to 3 in dominant epistasis to 9 to 7 in recessive epistasis. Note that some forms are even disappeared. If the recessive genotype of the hypostatic gene is active, not simply meaning that the dominant allele does not manifest, the number of phenotypical forms in the F2 changes. What is polygenic inheritance? How does it work? Polygenic inheritance, also known as quantitative inheritance, is a gene interaction in which a given trait is conditioned by several different genes, having alleles that may or may not contribute to increase in the phenotype intensity. The alleles may be contributing or non-contributing. There is no dominance among them. Polygenic inheritance is a type of inheritance, for example, of skin color and stature in humans. Considering a gene species of animals in which the length of an individual is conditioned by polygenic inheritance of 3 genes. For the genotype having only non-contributing genes, A, B, AA, BB, CC, the basic phenotype, for example, 30 cm, would be emerge. Considering also that for each contributing allele, a 5 cm increase in the length of the animal is added. So, in the genotype having the contributing allele, capital A, capital A, capital B, capital B, capital C, capital C, the animal would present the basic phenotype, which is 30 cm, plus 30 centimeters more than added by each contributing allele, i.e. its length would be 60 centimeters. In the case of triple heterozygosity, for example, the length of the animal would be 45 centimeters. That is the way polygenic inheritance works. What is the most probable inheritance pattern of a trait with Gaussian proportional distribution of the phenotypic forms? If a trait statistically has a normal distribution of its phenotypical form, it is probable that it is conditioned by polygenic inheritance, quantitative inheritance. The quantitative inheritance, the effect of several genes add to others, making it possible to represent the trait variation of a given population in Gaussian curve, with the heterozygous genotype in the center, i.e. appearing in large number, and homozygous in the extremities. How to find the number of pair of alleles involved in polygenic inheritance using the number of phenotypical forms of the trait they condition. Considering P, the number of phenotypic form, and A, the number of involved alleles of the polygenic inheritance. The formula P is given by 2A plus 1 is then applied. Many times it is not possible to determine precisely the number of phenotypic forms, P, due to the multigenic features of inheritance, since often the observed variation of the phenotypes seem to be a continuum, or the traits suffer environmental influence. Why is sex-linked inheritance an example of non-Mendelian inheritance? Sex-linked inheritance is a type of non-Mendelian inheritance because it opposes Mendel's first law, which postulates that each trait is always conditioned by two factors or alleles. In non-homologous region of the sex chromosome, the genotypes of these genes contain only one allele, even in the case of XX karyotypes, i.e. in women, one of the X chromosome is inactive. What is the mitochondrial inheritance? Mitochondrial inheritance is the passage of mitochondrial DNA molecule to the offspring. All stock of mitochondrial DNA an individual has have come from the mother and maternal grandmother and maternal great-grandmother, and so on. Since mitochondria are inherited from the cytoplasm of the egg cells that later constitute the cytoplasm of the zygote. There are several genetic diseases caused by mitochondrial inheritance, like Leber's inherited optic neuropathy that leads to loss of the central vision of both eyes, and Kearns cell syndrome, a neuromuscular disease that causes ophthalmoplegia and muscle fatigue. Mitochondrial inheritance is an excellent means of genetic analysis of the maternal lineage, just like the Y chromosome is an excellent means of the study of the paternal lineage. Thank you very much for participating in non-Mendelian inheritance. Please do like, share, and subscribe.